Hello, I am Jessica Damoiseaux. I'm an assistant professor in the Department of Psychology and Institute of Gerontology at Wayne State University. And today I'm going to talk about resting state functional MRI. I have nothing to disclose. So just a few key terms that I'll be using during this presentation. So the first is functional MRI. So functional magnetic resonance imaging. This is an imaging method that uses the blood oxygen level dependent, also known as the BOLD signal, as an indirect measure of neural activity. So then resting state fMRI. This is fMRI data that is collected in the absence of an external task stimulus. The default mode network, a set of brain regions that is more active during rest than task. And functional connectivity, which is a measurement of the association between BOLD fluctuations across different brain areas. The outline of my presentation, I'm going to start with some learning objectives. Then I'll give a brief introduction into the sort of history of resting state fMRI. I'll cover a couple of measures of resting state fMRI, and then I'll discuss the application of functional connectivity in aging and Alzheimer's disease. So at the end of this presentation, you should be familiar with the development of resting state fMRI as a field, also know different resting state fMRI measures and their applications, and understand the differences in resting state functional connectivity with older age and in Alzheimer's disease. So a historical perspective of resting state fMRI. So in 1995, Biswell published this paper, and this is the first study that showed that you can use functional connectivity in resting state data to look at this brain network. So what he did here is that he extracted here the signal from a voxel in the left motor cortex, and with that signal he correlated that with all the other voxels in the brain, and that gave him this network of brain regions, which really nicely shows the whole sensory motor cortex, or motor cortex, both left and right hemisphere. And this pattern that you see here is very similar to what you would see, what you see here in this part of the image, when you had participants perform a task. So this is a finger typing task, and here you see also the really nice bilateral motor cortex. So Biswell showed for the first time that you can also show this pattern with resting state fMRI data using this functional connectivity method. So and his work had been replicated after that. So you see that in this study they showed similar results, also looking at the motor cortex and using functional connectivity with resting state fMRI data to really show nicely this whole sort of network of the motor cortex when people were actually not doing any finger tapping task. And Hampson in 2002 also showed that you can do this with other brain areas and brain networks as well, as you see here with the language areas, where you also see that with resting state fMRI, if you do functional connectivity, you see this nice pattern of bilateral sort of language areas showing up in a network. So around the same time as this research into functional connectivity methods was developing, there was also another observation made here by presented here in this paper by Schulman, where he looked at task data. This is actually with PET data, and this is a meta-analysis of several PET studies that had people perform a task while they were in the PET scanner. And what Schulman noted here was that across these different task paradigms, he consistently noted that the same set of regions was less active during the task condition. So there was decreased activity in the task condition in this set of brain regions. And this sort of network of brain regions includes the medial prefrontal cortex, posterior cingulate, and lateral parietal areas. So, and a couple of years later, Mark Rako interpreted this sort of deactivation or less activity during task, as that there must be during sort of a rest condition when you're not focused on anything external, there must be a sort of an organized baseline default mode of brain function. So this is really the first time that this term, the default mode network, was mentioned. And this set of brain regions, the default mode network, is sort of thought to be involved in memory function and self-referential processing. And then a couple of years later, Michael Gracious combined sort of this approach of functional connectivity that was first published by Bharat Biswal, and then also this idea of this default mode network in this study, where he placed the seed region, or he extracted the time series from a region here in the posterior cingulate cortex, and then correlated that with the rest of the brain to see if he would, get this whole sort of default mode network. And indeed, he showed that by looking at the functional connectivity of the posterior cingulate, you can nicely, get this whole default mode network. And subsequent studies have, continued to look into these sort of resting state networks, and found that there are several of these networks that you can see during resting state. And this is a study of my colleagues and I that showed that we actually see sort of 10 of these networks that are consistent across subjects and also consistent across different sessions. So in these different resting state networks that we observe here include this default mode network. So this is the one that, by Mark Rakel and Michael Gracious that they pointed out. But we also see other areas, as you can see here in yellow, which are more visual areas, so lateral visual network, more medial visual network. And, for example, also executive control networks. So, and these are actually a set of brain regions that are usually activated by task performance, so based on different tasks. So you see that during rest you can find all these patterns, all these resting state networks present in the brain. So these networks, you don't only see them during rest, you can actually also see them during task fMRI data. But in this presentation, I'll mainly just focus on the functional connectivity using resting state fMRI. So, and now that we knew more about these resting state networks, we started applying some of these networks and measures that we can take from these networks to look at differences in across participants and maybe patient populations. So there are a couple of commonly used measures to measure functional connectivity. So I'm going to briefly discuss 3 of them. So I'm going to talk about seed-based correlations, data-driven approaches such as spatial ICA, and also whole brain network measures using graph theory. So the seed-based correlations, so this is actually the approach that Bharat Biswal presented in his 1995 paper. So just to go over that briefly again. So what you would have a seed region. This could be a larger area. It could also be a voxel. You extract a time series, from this area, and then correlate this time series either with all the other voxels in the brain or with predefined target regions, as you can see here. So you would do a correlation between these time series, and you can do that with different areas throughout the brain. And when you see that there's significant correlation between these areas, you can say that you can speak of a network of areas that are connected. And so in here you would see this is like the default mode network is one network of connected brain regions. So with this approach, you would have to select a seed region that you start from, and then you can see what other regions are significantly correlated with that. Another approach is to use a data-driven approach, such as spatial ICA. So the difference here is that you don't have to predefine a seed region that you start with, but you can really just let the data come up with a decomposition, into networks. So what spatial ICA does, and very briefly, so if you would have your fMRI data, as you can see here, you would have your space, all the voxels, and all the time points that you scan. So you would have all your fMRI data. So then this data set is decomposed into a set of time series, as you can see here, and a set of spatially independent maps. So here you would see that is really just this, spatial maps, and then separated in these independent components, as you see here. So what is done here is really, you impose spatial independence on these maps. So what that looks like is what I showed you earlier. So this is the previous study that I performed and that I showed you. So this is an example of using ICA to decompose the data, and you would get all these different independent components here. And you can nicely see that these different independent components really have these nice networks that you can also identify using this seed-based correlation approach. So then the last measure that I want to discuss is whole brain network measures, for example, using graph theory. So what you would do with this approach is that you have your brain data, and you can go two ways. You can even look at structural brain networks. We're more interested here in functional brain networks. But you would parcellate the brain into different regions, which you can base on anatomy. You can also base it on differences in function. But you would parcellate the brain so that you get multiple sort of nodes that you get collect data from. So all these different nodes, you extract a time series. So this is pretty similar to the seed-based correlation. You extract a time series and correlate that with the time series of all these other nodes. So what you would get here is like a whole network of all these different nodes and the correlation between these nodes and whether or not they are connected. And then you have this functional brain network, and then the graph theoretical approach is sort of, as you can see simplified here, is different measures that you can take to look at the efficiency, for example, of this network. So for example, you can look at what is the average path length between different nodes. And so for path length, you really just look at the number of edges between two nodes. For example, from this node all the way to this node, you would have 3 edges here. You can also look at clustering. So do we see that there are specific clusters within this network? And all of these measures are sort of a measure of whole brain sort of efficiency of your network, which you can then also use to compare efficiency between subject populations, for example. So now, how do we apply these measures? So I'm interested in studying aging and Alzheimer's disease. So I'm going to show you some examples of the effect of age on functional connectivity. And what we sort of know now in about 10 years of research looking into this effect, we kind of see this main pattern that we see lower connectivity in the default mode network in older adults compared to younger adults. And a couple of the first studies looking into this was one of my own studies that looked at default mode network functional connectivity. And we noticed that specifically in these two, so these are two sort of subsets of default mode network, and in this anterior default mode network and this posterior default mode network, we saw lower connectivity in older adults compared to younger adults. And around the same time that this work was published, there was another study that found a similar effect. And they used a different approach. So we used an independent component analysis, so a data-driven approach. Andrews Hanna, they used an approach, a seed-based correlation. So they extracted the time series from this posterior cingulate and from the medial prefrontal, and they correlated these time series. And they also found that compared to the younger adults here, that the older adults showed lower connectivity between these nodes of the default mode network. And they also found that within the older adult group, that the older age was correlated with lower connectivity. So these findings have been replicated in the past couple of years. And even though there are some studies that report differences in other resting state networks in addition to the default mode network, these effects in the default mode network have been replicated most consistently. So more recently, I've also been interested in looking a little bit more specifically into this default mode network connectivity with age. And I was interested in the involvement of the hippocampus in this default mode connectivity, and specifically if there was any difference between posterior hippocampal connectivity to these default mode network nodes and the anterior hippocampus. And indeed, we did find a difference in the connectivity of these two hippocampal regions, with other DMN regions, in that the main effect of age was with the posterior hippocampus. So you see here in red, in all these scatter plots, in red is the posterior hippocampus connectivity. So here the connectivity of the posterior hippocampus to the medial prefrontal cortex. So again here, posterior hippocampus to medial prefrontal cortex. You see here declines with age. And this is a lifespan sample, so it's really from younger adults all the way to older adults. And you see that connectivity gets lower with older age. And this is significant for the posterior cingulate, for the posterior hippocampus, but not for the anterior hippocampus. And similarly here, for posterior hippocampus to posterior cingulate, this is also significantly decreased with older age, and also for the posterior hippocampus with the lateral parietal areas, which you see here in purple. So overall, this result shows us that it's mainly the posterior hippocampus that is part of this default mode network, and that shows a similar decrease with older age. So then, if we really want to talk about real changes in functional connectivity, you would have to do a longitudinal study. These previous studies were cross-sectional studies. So in this longitudinal study, they looked at 3 networks, so the default mode network, as you can see here, executive control network, and the salience network. And across these 3 networks, you see that with time, and this is 4 years, a span of 4 years that you see in these spaghetti plots. So over 4 years of time, you see a decrease in within-network connectivity in these older adults. So you can see quite consistently that almost all of these older adults show that with time, the functional connectivity in the default mode network decreases. You also see that the older adults seem to have lower connectivity than the younger older adults. So this was significant for the default mode network, also for the executive control network, where you also see that over the 4-year span, you see a decline in functional connectivity within the executive control network. For the salience network, this was not so clear, so it seems to go in multiple directions here. So then they also investigated between network connectivity, so not just within network, but also between network. And they showed here the between network connectivity of the executive control network and the default mode network. And they found a U-shaped trajectory of changes here, so that mainly the younger older adults, they showed over time, they showed a decrease in between network connectivity, but the somewhat older adults showed an increase in between network connectivity. So this is interesting to see that there may be sort of a change, in the trajectory, of sort of changes within and between network connectivity, so that the between network connectivity may initially decrease and maybe later increase again. And so we see that, with aging, we mainly see that these default mode network areas show changes with older age. So what I'm also interested in is looking at the effect of functional connectivity changes in Alzheimer's disease. And interestingly, what we see here is that similar to what you see with normal aging, you also see that in Alzheimer's patients that there's lower default mode connectivity versus healthy older adults. So again, the same set of brain regions seems to be affected with Alzheimer's disease here as well. And this was initially shown by Michael Gracious in 2004. As you can see here, that in the default mode network, that there was lower connectivity in Alzheimer's patients versus healthy older adults. And this result has been replicated several times, which was nicely reviewed in this article in 2014. So when I started working with Michael Gracious, we initially wanted to see if we could also replicate his previous finding of lower default mode connectivity in Alzheimer's disease in a new data set. So we examined this, and we indeed found, and here looking at the posterior default mode network, so we actually, again, here sort of split the default mode network into these several subnetworks. And if you look at the posterior default mode, we found the same effect as Mike Gracious had found earlier, so lower default mode connectivity in Alzheimer's patients compared to healthy older adults. But when we looked at these other default mode networks, so the more anterior network and also sort of a ventral network, we actually observed higher default mode connectivity in Alzheimer's patients. So this was a bit different than we expected, because most of the previous research just showed generally a lower connectivity in the default mode network. So we were interested in what sort of could explain this, or what mechanism is going on here with this higher connectivity, and also what would happen if you would follow these patients over time. So that's what we did with this study, and we followed these patients 3 years later. We had them back, and 3 years later, if we look at the change, from baseline to these, the follow-up, we actually see that across all these networks. So we looked at the anterior and the posterior default mode network and this ventral default mode network, that we only saw decreases. So it was only, decreased functional connectivity in these areas. There was no increases anymore. And we also looked at this sensory motor network, which initially was a control region. So in our baseline study, we did not expect any differences between healthy older adults and Alzheimer's patients in the sensory motor network, and we indeed did not find anything. But then over time, so 3 years later, in Alzheimer's patients, we also saw a decrease in the sensory motor network. So at this time, these patients were more severely demented. So they had sort of an average MMSE of 20. So you could see that in these patients, there seems to be just an overall decrease in functional connectivity across multiple brain networks. So, and this pattern of changes that we saw this initial increase and followed by a later decrease, was also found in this study by Bai in 2011. They looked at patients with amnestic mild cognitive impairment, which we consider to be a precursor of Alzheimer's disease. And what they found here was that actually at baseline, they found increases in connectivity compared to healthy older adults in this posterior cingulate area, precuneus area. And then after follow-up, a couple of years later, they saw that the same area that initially was increased was now decreased, showed decreased connectivity. And they saw increases in these more frontal brain areas. And the pattern that you see here is more similar to the pattern that we saw in our baseline Alzheimer's patients. So this sort of led us to believe that maybe there is this sort of pattern of changes that you see in Alzheimer's disease, where you initially see hyperconnectivity in an area which precedes this hypoconnectivity of the default mode network. As you can see, this follow in this MCI baseline follow-up, AD baseline, and AD follow-up. So, and this trajectory of changes is not random. It actually seems to follow this trajectory of pathological changes that was observed by Brack and Brack. That really shows that sort of it seems to start, the pathology seems to start more in the medial temporal lobe and then spreads, through actually areas of the default mode network, so more posterior cingulate, as you can see here, and then spreads more to the front of the brain in a later stage. So it may be that we really see sort of that the start of pathology is actually illustrated by this increase in connectivity and then later followed by a decrease of connectivity. So to summarize, we went over a little bit of history of the resting state functional connectivity, and I showed you that in 1995 was the first study by Brad Biswell to look at functional connectivity of the motor cortex, and then in 2001 Mark Rakel coined the term the default mode network. So then we went over a couple of measures of resting state functional connectivity, so seat-based correlations, data-driven, spatial ICA, and more whole brain network graph theoretical measures. And then I showed you some examples of functional connectivity research in aging and Alzheimer's disease, and that we find that really the difference in the default mode network seems the most apparent with older age and also in Alzheimer's disease, and that these disease-related decreases in functional connectivity, that they actually may be preceded by increases. And this is important, especially if we want to look at Alzheimer's disease in an earlier stage. So for potential early detection of Alzheimer's disease, we may actually have to focus more on these increases in functional connectivity than the decreases, because this may be the first sign of abnormal brain function. So this concludes my presentation. If you have any questions, feel free to contact me by email. Thank you.